Hello, my name is João Victor, and today I'll present a study that's being developed with the supervision of Professor Fernando Mantelatto, Master Jennifer TELES, and Professor Harry Boss, and it's entitled Preliminary Insights on Genetic Data about Brazilian Decapod Red List. The selection of the species was executed taking into account this red book. So, this book lists frequent crustacean species according to their category and also provides information on the species biology, ecology, and geographic distribution, in addition to citing possible conservation actions and necessary research. So, for this work, species were selected from these four categories: critically endangered, vulnerable, and near threatened. Here are some decapod species that are threatened in Brazil. So, here we have examples of freshwater crustaceans like the Aiglas and Parastacus. They are affected mainly by the degradation of freshwater environments. We also have lobsters of the genus Panulirus, which suffer mainly due to illegal fishing. And in this last picture, we have the crab Ucides cordatus that lives in mangrove environments. The main objective of this study is to develop a genomic database of endangered decapod crustaceans in Brazil. For this to occur, new sequences will be generated for some species of decapods, and later it will be possible to analyze the richness and phylogenetic diversity of these species, and find localities and taxonomic groups that are most threatened. From this, it will also be possible to suggest additional conservation measures for the most vulnerable species and environments. For the execution of this study, a data survey was carried out on platforms such as GenBank and Bolt Systems, and only sequences present on one platforms and that contain an excess number were considered deposited. After this data survey, some of the missing species were already in the CDB, and others were borrowed from other carcinological collections. After that, laboratory procedures were performed, the editing of sequences were executed through Genius, and statistical analysis performed in R. Two phylogenetic trees were created through the software R1 for each gene, and these trees expose the species that have already acquired sequence, and these species highlighted in bold were the ones that have the sequence generated in the present study. From the data survey, some graphs were elaborated that exposed the lack of genetic data in the studies of threatened species. Note that for both genes, most of the listed species do not have a deposited sequence. Furthermore, even for those with deposited sequence, most of them are in the near threatened category, which is the least critical compared to the others. Also, the nucleotide diversity of each infraorder was calculated, which is an indicator of genetic diversity. In the results, it highlighted the value for the infraorder Anomura, which may be related to the fact that all species in this group belong to the genus Aegla. This genus is characterized by a high degree of endemism and low genetic diversity, and these factors contribute to the high number of Aeglas listed in the Red Book. Here are some pictures that illustrate the environment that the most of the species of Aegla inhabit. So here we see examples of São Pedro and Claraval in lotic freshwater environments, but there are some species that live in lentic waters or inside caves too. A mapping of the richness and phylogenetic diversity of these species in the Brazilian territory was also carried out. In both maps, there is a concentration of these species in the south and southeast regions, and also in coastal environments. These localities in south and southeast are similar to the regions that occurs the distribution of the genus Aegla in Brazil. And the richness of these species in coastal environments represents many species that suffer from mangrove degradation and fishing. With all that has been present, we note a scarcity of genetic data for endangered decapod species, and this scarcity also reveals how this data is not taken into account in conservation studies and as a criterion for assessing the risk of extinction. In addition, it's also evident the predominance of genus Aegla among the threatened species, which should receive attention due to its high endemism and low genetic variability. Among the most threatened localities, we can highlight mainly coastal regions, which suffer from environmental degradation due to a high anthropic occupation, and also freshwater localities in the south and southeast, where aeglids are distributed. Here are the references used to support this research in main aspects of this presentation, and this project is supported by the following research agencies that I would like to thank all.